because you've got a case where they evolved two different solutions to this problem: homologous recombination and non-homologous end joining. Homologous recombination is you go find the other copy of the DNA that you're dealing with in the cell and copy a good copy from that one, and you replace the old DNA. Non-homologous end joining is, I don't care, it's broken, and stick the thing together. Double-strand breaks are very, very problematic, because if you've got a strand of DNA with 10 to 20 bases apart, broken on opposite strands, so you can think of it like this: you've got a break over here, and you've got a break over here, that will melt in the normal environment. And so now you've got free-floating DNA ends flopping all around all over the place. And there are all kinds of problems this can create. Loss of the chromosome so that it has no centromere. You can't divide and separate out the chromosomes if you've got no centromere. Loss of the telomeres on both of the fragments. Now you've got, uncapped DNA floating around in the cell, which the cell doesn't like, because the cell considers that typically to be a virus and will try to digest it and get rid of it. And then you have also the loss of the gene. If the break happens in the middle of a gene, the gene is broken. So chromosome fusions, if it's misrepaired, this results in chromosome translocations, which are bad because a lot of them can cause cancer. So they are an extremely high priority for the cells to repair. You don't want a lot of double-strand breaks. Failing to repair causes the induction of apoptosis, so the cell will commit suicide if there's too many of these. One of the ways species may evolve longevity is by becoming stricter and stricter about counting the number of double-strand breaks in the cell and deciding to commit suicide if there's too many to put back together. They've done experiments with painting mouse chromosomes, and they find that mice that are older, that have had problems with double-strand break repair, may have put even 3 chromosomes back together improperly. So they potentially could have lost fragments of those chromosomes, and then the 3 that are stuck together could have all kinds of problems with them. Homologous recombination can be preferred in simple animals that have little duplicated or junk, so-called junk DNA. And that's because when you're trying to line up 2 strands to find the partner strand for the strand that's broken, you have a low probability of finding a matching strand which is in the wrong place. While non-homologous end joining is just joining things together. It's more preferred in complex species because doing homologous recombination in species with a lot of junk DNA can potentially result in copying in the wrong DNA from the chromosome to where you're trying to repair. Now, over the last decade or so, we've now found out that this is what's involved in repairing chromosome breaks using non-homologous end joining. And so the way this works, and Ralph likes to talk with his hands, so I figure I can do it too. You got the break. So these 2 things are floating around here, all right? First thing you got to do is say, don't digest that DNA. It's important. I need it, okay? So KU7D and KU86, otherwise known as XRCC6 and XRCC5, grab onto the DNA on the end of each of these and say, okay, all right, we're the broken ends here. Then PRKDC comes along, and PRKDC is a kinase, and so it's responsible for saying, oops, we've got a problem here, guys. This is the Houston, we've got a problem enzyme. And so it will in turn talk to P53, which is responsible for saying, how many of the breaks do we have here? If we got 3 of them or 4 of them, we have a real problem. And then it will tend to commit suicide. ATM and ATR, on the other hand, saying, well, we got a break, maybe I can deal with this with homologous recombination. That's one of the things involved. Now here, if you really want to point to genes that cause aging, these are your 2 culprits. Because the Artemis gene, which was only discovered, I think, in the late 90s or early 2000s, but it was previously known because it causes severe combined immune deficiency. So it was thought for a long time that this was an immune system gene, but it also turns out that it's involved in double-strand break repair. It's involved in the immune system because this nuclease creates the variation that's necessary for the formation of the immune system. The variation that you need in antibodies, the variation that you need in creating the MHC receptors. And it also has an endonuclease, because loose DNA like this can bend around on itself and bind to it and bind to itself. And if that happens, then you want to cut that off completely, so you've got a free end you can play with. And then some of these, they still don't know what these do. This one may be quality control, because if you can think about two strands, you can put them together like this, or maybe they should be going together like this. Now, if you do it like this, it's bad. If you do it like this, it's better. And then, but these two parts of these two genes are highly conserved, going all the way back to some of the most primitive organisms. Perhaps even, I think I looked at sea urchins once, so that's how far back they go. That's how important they are to maintaining the structure of the DNA. DCLRE1C, actually these are all highly conserved. The only case I've found currently, I believe, is in C. elegans, which only lives 7 days. Has lost the helicase off of the Werner's gene, I think, which it's only living 7 days. It doesn't care if it puts things back together. So now, proof of this. So here's an experiment that was done in Drosophila. One of the problems is, because these mutations, these double-strand breaks, are happening at random locations in the cell, in order to find them, you would have to go and sequence the individual genome of each cell. And we're currently not at that state of sequencing the DNA. But if you put a marker gene into the cell so that you can say, oh, if I've got lots of animals, I can select one where I know the mutations have occurred in that gene. And then you can, and because you know what the sequence of that marker gene is, you can sequence just that area. Researchers have done this in the early 1990s on the HPRT gene and the APRT gene by Richard Albertini. And that said, well, yeah, no, gee, somatic mutations are really happening. But those are the only two genes that it was studied in, because in the early 1990s there was no sequencing technology for all practical purposes. But since we have the sequencing technology now they can look at these and say, how did these change? Well, all right, this one's unchanged. That means that you happen to have a break where everything went back together very nicely, back to the original state. However, here, all these yellow ones are simple deletions. Oops. When we put it back together, that's a 1 base deletion, a 2 base deletion, a 3 base deletion, a 4 base deletion, and so forth. Now, it also turns out if sometimes it's gotten wrong and it puts it back together like this, now you can get an insertion, because this gets filled in and this gets filled in. And so it's possible to get the insertions here as well. And then these are the interesting ones, because these are deletions. These are the insertions, and these are the deletions plus insertions. So it's like, okay, we'll chew up a little bit, and then it still doesn't go back together, but we'll stick it back together and fill stuff in anyway. So you see there's all kinds of mutations that result from this particular form of repair. But it's better than letting the chromosome get lost or having to kill the cell off. It's like, well, yeah. And the choice you make in going to the kill it or save it at all costs perspective is the key aspect that's there. Now, okay, you can say, well, that's just Drosophila. You know, that only lives a month anyway, so who cares? The interesting thing is you can also find these in humans if you know where to look. And the sequencing technology has advanced far enough that we now have the two projects. One is at the Sanger Center in England, and the other one is at various centers in the US. And so what they have said is, we are going to start a massive project. We want to look at 20 major cancers, because everyone knows the story. Why can't the chemotherapy, why can't the radiation, et cetera, work against cancer? And the reason is because the genomes are all mutated in different ways. And so they're doing mutational analysis on the genomes. 20 major cancers, 20,000 tumors they're going to look at. That's a lot of tumors. That gives you a very statistically significant sample. They're looking at 600 plus genes. The first round they looked at 600. They said, that doesn't completely explain the problem. They're now going back and looking at 1,200, 1,500 of that order. People are providing a list of their next favorite gene or the next most likely candidate. Now, the interesting thing about this is that if you look at the results, you get all kinds. These are point mutations where a single base is changed. splice site mutations where the splicing of the gene changes, frame shift mutations, which is when you have shifted the register of a protein reading frame, which means all of the amino acids downstream from the mutation get turned into a different amino acid. And you've also got chromosomal aberrations. So one of the things that happens here is these and these and these all typically can alter protein foldage. So you get proteins which will never, ever fold properly. And that's not a good thing, because you'd like proteins to fold properly and do what they're supposed to do. And then the chromosome alterations can alter gene expression.